In the previous module, we saw the two ends of microtubules and actin are different. Different sequences of amino acids are exposed at either end. This also changes the ability of these filaments to add new subunits at the respective end. So let's talk about the growth rates at these two different ends. The structural polarity of actin filaments and microtubules is created by the regular parallel orientation of all the subunits which are making these filaments. The orientation makes the two ends of each polymer different. This difference has profound effect on filament growth rates. In absence of ATP or GTP hydrolysis, the free energy difference and therefore the equilibrium constant and also the critical concentration are expected to be same for addition of subunits at either end of the polymer. In structurally polar filaments, the kinetic rate constant for association and dissociation, which we refer to as K on and K off, are often much greater at one end rather than the other. If an excess of purified subunits is allowed to assemble onto marked fragments of the preformed filaments, one end shows preferential addition of monomers as compared to the other one. So the monomers are adding faster and that end elongates faster than the other end. If the filaments are rapidly diluted, so the free subunit concentration drops below the concentration. Remember in the graph we saw that our growth of the polymer or polymerization plateaus off, and at that the concentration at which the addition of new polymer is not changing is called the critical concentration. At that concentration, basically, if we go below that concentration, your polymer begins to disassemble or depolymerize. So the one end depolymerizes faster than the other. The more dynamic of the two ends of a filament where both growth and shrinkage are faster is called the plus end, and the other is called the minus end. Okay, so we have talked about this briefly when we were talking about looking at the ultrastructure of these polymers, plus end, minus end. So plus end grows faster, depolymerizes faster, shortens faster than the minus end. So here, let me show you an experiment which was done in Dr. Borasi's lab, who was one of people who supervised my PhD at University of Wisconsin. So here, if we add a stable bundle of microtubules, you can obtain a stable bundle of microtubules from core of a cilium. We'll talk about cilium later when we are talking about cytoskeleton. And it was incubated with tubular subunits under the conditions that favor polymerization. Microtubules grow faster at the plus end. This is the new growth. All you are seeing is this is the new growth versus the minus end. The growth is not quite as robust as it is at one end of these microtubules, which is basically the plus end. So here, this is graphic form of showing you that the two ends of microtubules have different abilities to add subunits, plus end, minus end. On microtubules, alpha subunits are exposed at the minus end, and beta subunits are exposed at the plus end. I hope that is clear from our discussion in the previous module. On actin, the ATP binding cleft on the monomer points towards the minus end. We saw that when we were looking at the ultrastructure there. Filament elongation proceeds spontaneously when free energy change, delta G, if you recall, for addition of soluble subunit is less than 0. So if delta G is less than 0, that's a favorable reaction. You can think of addition of monomer to polymer as a reaction, it being a reversible reaction. You can add monomers or you can remove monomers. It is a reversible reaction. So if delta G for addition is less than 0, the reaction will proceed and you will have assembly of monomers into polymers. If the concentration of subunit in solution exceeds the the critical concentration, that is when it happens that you start polymerizing these structures. When filament depolymerization proceeds spontaneously, the free energy is greater than 0, which makes sense. If delta G is a positive number, you will lose the subunits rather than adding them. A cell can couple energetically unfavorable process to the spontaneous processes. Thus, the free energy released during spontaneous filament polymerization or depolymerization can be used to do mechanical work, in particular push or pull, or you can also move a load, which happens when cells are dividing. You remember the spindle forms, and chromosomes are attached to these spindle fibers, which are basically microtubule fibers. So if chromosomes are attached to the microtubules and the microtubules begin to shorten, they start losing the monomers, the chromosomes will be pulled apart on the either side. Also, we know that microtubules, as we saw earlier, can be used to push membranes. Other examples, here I'll read off the slide. For example, elongating microtubules can push out the membrane, and shrinking microtubules can help pull the mitotic chromosomes away from their sister chromatids during anaphase. Elongating actin filaments help protrude the leading edge of motile cells at a different stage. I will also show you a video where you can see these cytoskeletal elements exerting force on the membrane, which results in either cellular change in cell shape, or it can also result in movement of the cell. So that's basically, we have seen that the growth rates at two ends of these filaments, the microtubules and actin filaments, are different.